This video will be talking about granulomatous inflammation. This is a high-yield topic for USMLE Step 1. Stay tuned till the end. Granulomatous inflammation is characterized by the formation of granuloma. So granuloma is basically aggregate of immune cells, primarily macrophages, which basically is surrounded by lymphocytes. Now these granuloma forms because the immune system want to contain the bacteria which are hard to eliminate. So immune system doesn't allow these bacteria to spread throughout the organ. They want to confine these bacteria into a particular volume. So that is how granuloma forms. These are basically macrophages which are trying to prevent these infections from spreading. So obviously we understood macrophages are the key drivers of this granuloma formation. So let us try to understand the process. So the first step is the initial encounter where the macrophage is trying to engulf the pathogen. In general terms, macrophage would engulf the pathogen and destroy them. But in some cases, for example, Mycobacterium tuberculosis, destroying these pathogens is difficult. Sometimes these bacteria clump together and overwhelm the macrophage. So in those conditions, macrophage gets activated, and these activated macrophages lead to formation of the granuloma. Now, when they are activated, there are certain cellular and morphological changes occur within these particular macrophages. And activated macrophages ensure that they basically prevent or confine these invaders into a restricted location. There are two types of macrophages broadly, but M1 macrophages are pro-inflammatory and they have central role in defense against pathogen, and M2 macrophages are anti-inflammatory in nature. Now in this particular granuloma formation, several activated macrophage cells are recruited, which prevents the bacteria to spread further. Eventually, these macrophages secrete pro-inflammatory cytokines such as IL-1, IL-6, etc., which attracts Th1 subtype of T helper cells. And all these cells clump together, form these granuloma. Inside the granuloma, there are certain changes in the macrophage structure. For example, several macrophages fuse to form a giant cell which appears as multinucleated cells. There are also activated macrophages. There are also lymphocytes found in this granuloma structure. Now let us understand the overall histology in a bit more detail. So here one can see the lymphocytes in the periphery of a granulomatous formation. One can see epithelioid altered macrophage cells. And one can also see giant cells which has several nuclei, which are basically fused macrophage cells. Now there are several types of giant cells. For example, Langerhans giant cell is one of the most common type of giant cell, and this is commonly associated with tuberculosis infection. So in this case, what is the pathological significance? This Langerhans giant cell, which exactly look like a horseshoe-shaped region, this is basically a pathological hallmark of tuberculosis infection. So from a lung biopsy or from other tissue biopsy, if somebody can notice this sort of formation, one can really suspect that there is an invasion of tuberculosis. There are other instances where the infective organism is not really a bacteria. It's basically some sort of foreign material which is not present in the body. Let's say some talc powder or some crystals that might lead to this sort of granuloma formation. That is known as foreign body giant cell formation. In this case, there are also multinucleated cells which are the giant cells, but the nucleus are arranged randomly. It's not like the Langerhans cell where there is a characteristic peripheral arrangement. Here the arrangement is quite random, and basically they form due to foreign body exposure like tal, sutures, etc. Now let us try to appreciate the morphology of the giant cells. For example, in Langerhans cells, nucleus are peripherally organized. In foreign body giant cells, the nucleus are haphazardly organized. There are also situations which are known as Toton giant cells. There are tumor-associated giant cells associated with anaplastic cancers or Hodgkin lymphomas. For example, Red Steinberg giant cell is associated with Hodgkin's lymphoma. Then there is osteoclastic giant cell. All of these are collectively known as tumor associated giant cells. Now, since you are more familiar with the histopathological features, now let us look at our original section. Here you can see the core of these granuloma actually comprises of macrophages and multinucleated giant cell. Surrounding the core, there are several other immune cells such as mostly lymphocytes. There could be also fibroblasts which are associated with tissue regeneration. Now let us talk about the common features of granuloma formation. There could be basically agents or pathogens which can lead to granuloma formation. This includes sarcoidosis, tuberculosis, leprosy, brucellosis, syphilis, and also lymphogranuloma venerum, which is basically a sexually transmitted disease. All these diseases might lead to granuloma formation. At this point, it's important to note that granuloma formation and granulomatous infection is very different from the chronic granulomatous disease or CGD, because CGD is basically an inherent disorder. In this case, a gene is mutated due to which the neutrophils are unable to destroy the pathogen. So neutrophil involvement is heavily associated with this particular disease. Neutrophil can kill any bacteria with the help of reactive oxygen species burst or oxidative burst. And these reactive oxygen species are generated by an enzyme called NADPH oxidase, which is encoded by the gene. So obviously there is a mutation in this gene which lead to reduced production of ROS. That is why even if the pathogens are engulfed, they are not destroyed. This could be X-linked or even autosomal as well. Now, generally, whenever there is a pathogen engulfment, the oxygen is converted into superoxide radical with the help of NADPH oxidase. Then superoxide dismutase convert it into H2O2. Eventually, myeloperoxidase convert into HOCl radical. And these free radicals are actually damaging to these pathogens. They take part in elimination of these pathogen. It's important to note that these free radicals are also regenerated inside the neutrophils via specific pathway. Now, how does ROS-can damage any protein or any bacteria? So ROS can alter the lipid composition. It lead to lipid peroxidation, thereby damaging the membrane. It can also damage or structurally alter the proteins. It can lead to DNA damage and problems in DNA repair. This is how it kills the bacteria. In case of CGD, these aspects are abrogated because NADPH oxidase, which is supposed to produce ROS, is inactive. And that is why even if the neutrophil is engulfing it, it cannot degrade it. That is why they form clumps, which is again a granuloma formation. But in this case, neutrophils are the key molecules which are associated with these granuloma formation, along with macrophages, dendritic cells, etc. Now, patients that are at high risk of infection by these patients which has CGD, they are at very high risk of infection by Staphylococcus aureus or Aspergillus, etc. Now, the symptoms include enhanced infection by Staphylococcus aureus, fungal infections like Candida, Aspergillus, etc. Symptoms might include persistent fever, pneumonia, skin infection, GI issues, etc. Now, there are many complications that could be associated with CGD. This might be, some of these are obstructive uropathy, hepatosplenomegaly, and also inflammatory bowel disease, but these are secondary. So now we can understand clearly that granulomatous infection and granuloma formation or CGD is very different. So I hope this video was useful and informative. If you like this video, give it a quick thumbs up. You can support our channel using super thanks. See you in next video.